Cambrian explosion refers to the appearance of most major animal body plans in the fossil record, estimated at 540 million years ago. It was a phenomenon encompassing the dramatic appearance of diverse metazoans with biomineralized skeletons, increases in complexity, behavior, and biodiverse marine ecosystems never seen before. This chapter of history laid the foundations for animal evolution. Fossils appear over 20 million years in a rapid burst during the early Cambrian, hence the term explosion. Time-equivalent environmental changes such as vast increases in oxygen are also considered important extrinsic causes. Rapidly accumulating genomic data alongside the fossil record supports the radiation of bilateral crown groups in the latest Precambrian Ediacaran, around 513 million years ago. Crown brachiopods and mollusk species also radiated in the early Cambrian, around 522 million years ago.
Are critical to calibrating the age of branching events in phylogeny, and most can be placed into a stem or crown group of living phyla. Although simple traces of bilateral animals were found to be present by 560 million years ago, Metazoan trace fossils reflecting a rapid diversification of body plans didn't appear across a broad range of marine environments until the earliest Cambrian. The Burgess Shale and Chengjiang provide excellent Cambrian fossil deposits, including biotas that indicate all major Metazoan clades, along with vertebrates that appeared 518 million years ago. Examples of Burgess Shale fossils can be seen to the right. Biomarker studies of organic materials, often lipid particles extracted from such fossils, have been analysed in terms of their molecular content to provide insight into age. One reason for the explosive number of fossils from the Cambrian period is the result of biomineralization in the formation of the skeleton. This is a major stepping stone from the soft-bodied organisms that existed before this time, causing important shifts in geosphere-biosphere interactions, along with the ecology of the Ediacaran Cambrian progression. Mineralization offers competitive advantage, support for larger body sizes, including protection from the ever-evolving predators of the Cambrian Sea. It may be a result of increases in O2 and alterations in seawater chemistry, and resulted in some strange-looking creatures such as hallucigenia, which can be seen here. Tissue mineralization appears independently in different lineages, with all biomineralized taxa preceded by soft-bodied organisms. Murdoch proposed the formation of the skeleton may originate from a conserved genetic toolkit, explaining how so many clades form the ability to produce skeletons.
Tracing GNRs across metazoan phylogeny is crucial for understanding the diversity of body plans that occurred through time and the Cambrian. Whole genomic sequences demonstrate that only a fraction of these genomes constituted developmental GNRs controlling body plans, and are therefore considered a necessity for early metazoan diversification. The genome evolution across the metazoan tree shows important findings, such as that homeotic genes used in cell type differentiation and patterning in metazoans were already present in choana flagellates. For example, sponges contain large numbers of genes otherwise found only in new metazoans. Losses, gains, and duplications of genes dynamically evolved and have unbalanced distributions across the tree. Duplication rates higher at deeper nodes leading to metazoans, and the gains progressively decreased as the animals diversified into clades during the Cambrian. Deuterostomes and ecdysozoans in particular had no significant gene gain, but widespread differential gene loss. These show examples of early gene co-option and demonstrate that the dynamics of gene gain and loss shape vast morphological disparity amongst animals.
It has been proposed that the diversification of specifically the ANTP class homeobox genes, which encode for various transcription factors involved in body patterning, caused an extensive convergent diversification in species and the possibility for neofunctionalization. These genes increased in number in the bilaterian stem lineage and throughout the Cambrian explosion. The gene duplications produce a large array of ANTP class genes, including 3 distinct gene clusters, NK, HOX, and para-HOX. NK genes are expressed predominantly to pattern the bilaterian mesoderm. HOX genes encoded physiological position along the central nervous system, and it is thought that the para-HOX genes originally specified the mouth, midgut, and anus of the newly evolved through gut. A vast amount of such genes are conserved today. For example, orthologs of the NKL gene, NK2.5, plays a conserved role during cardiac development in modern insects and mammals. On an epigenetic level, it is interesting to note that, as a general trend, the level of DNA methylation increased with the evolution of structural complexity during the Cambrian. This function is absent or extremely rare in invertebrates such as Drosophila or C. elegans. The significance of DNA methylation to the evolution of developmental complexity lies in its role in the management of developmental pathways. Therefore, alterations of this epigenetic control can also cause alterations to developing structures. An accumulation of alterations could lead to, or certainly affect, divergence.